This video explores problems that may be holding you back in your understanding of ATP. It's meant for someone who's been introduced to the basics of the topic. I've tried to learn a lot, but I'm sure there's still gaps in my understanding. So if you find some, let me know. You'll also need to grab a piece of paper. Problem number 1. You think that ATP's functions rely on a phosphate group being flung with force. Draw a picture of ATP. Did you include the spring that some drawings of ATP have? The idea of the last phosphate group being attached by a spring really misled me. The repulsion between phosphate groups are not irrelevant to how ATP works. ATP is a good phosphate transfer because of how weak that bond is, which is in large part due to repulsion. But the repulsion does not fling off phosphate groups in order to transfer phosphate or to make your muscles move. Transferring phosphate, which is how ATP powers what would otherwise be unfavorable reactions, happens in the context of an enzyme where no flinging is required. Problem number 2. You think that enzymes catalyze reactions by snapping into a different shape. At some point, I forgot how enzymes work and thought that they involved things smashing together. Take a second to write down how they help reactions happen. Enzymes put reactants into an orientation and position that allows them to undergo a reaction. Enzymes can stabilize reactants through non-covalent interactions like hydrogen bonding and electrostatic forces, and might even provide or take a proton or two from the reactants to help the reaction occur. Problem number 3. You think that the forces present in reactants solely determine whether a reaction occurs. Write down what factors you think influence whether a reaction happens. If ATP is positioned correctly with another reactant by an enzyme, they can react and end up in a lower energy state, which is related to them being more stable than before. If the reactant repels phosphate less than ATP, it's likely that the phosphate group will transfer. Now, the other molecule has a phosphate group that it wants to replace with something else. Wouldn't it need to repel the phosphate group to get it off, like ATP did? Pause and write your thoughts. No. Think about the products. Phosphate is a great leaving group because when it isn't attached to anything, it has resonance. Resonance, where electrons are delocalized across bonds, makes a molecule really stable. It's not just the reactant's internal forces that determine whether a reaction happens. It's whether the products are more stable than the reactants. Under many circumstances, a loose phosphate and molecule X with Y attached have lower energy than a loose Y and molecule X with a phosphate attached. This is how ATP makes unfavorable reactions happen. Pause and fill in whether these would be positive or negative. Then explain how ATP allows what would otherwise be unfavorable reactions to occur in the cell. Problem number 4. You think that molecules magically know what rearrangement will have the lowest possible energy. How does the Gibbs free energy of products, which are only hypothetical futures of reactants, affect if the reaction proceeds? It might seem like it, but reactants don't see into the future. When reactants are in a stabilized transition state, there's a lot of movement of electrons, and bonds will form and break as they move around. When the molecules formed have lower potential energy than before, that energy is converted to kinetic energy, which means more jostling, that gradually dissipates to the surroundings as heat. If the molecules have enough kinetic energy, endergonic reactions are possible, but they become less and less common as heat continues to dissipate. All of these back and forth changes happen in a split second and mean that stabler products are more likely to form. Problem number 5. You think that coupled reactions happen separately by space or time. A common example of a coupled reaction is the phosphorylation of glucose in glycolysis. I don't think it's typically made clear enough that neither of those components, here labeled 1 and 2, happen. Instead, the example is just to show that glucose would not be able to be phosphorylated from a loose phosphate ion. So in order to get phosphorylated, the cell has to use an ATP molecule. Another reason I find it confusing is that I'm not sure that this reaction occurs in order to power subsequent unfavorable reactions. Instead, it's typically emphasized that the phosphate transfer traps glucose in the cell. A better example of ATP transferring a phosphate to power a reaction is the synthesis of glutamine by the enzyme glutamine synthetase. The transfer and replacement of the phosphate group happen as separate steps within one enzyme. Here, you might still hear people say that ATP hydrolysis is coupled to the addition of an ammonium ion to glutamate. But remember that ATP is transferring a phosphate and doesn't react with water in this case. Problem number 6. You think that all of ATP's roles depend on transferring phosphate that is later replaced. ATP doesn't just make unfavorable reactions happen. Pause and try to write down some of the other things that ATP does. To name a couple, it also transfers phosphate groups to proteins to slightly change their shape, which can activate or inactivate them. Sometimes it doesn't transfer phosphate at all. For instance, it undergoes hydrolysis to power ion pumps. Fill in the blanks about the mechanism discussed in the video. ATP blanks phosphate, and then the phosphate is blanked. The answer would be ATP transfers phosphate, and then the phosphate is replaced. Thank you so much to the contributors to the Biology Stack Exchange, where I asked many questions. Feel free to comment on this video with any corrections, curiosities, or confusions. I personally still don't understand how muscles and ion pumps are powered by ATP. Also, have you always been interested in being interviewed? Contact me if so.